Hi everybody, my name is Mary and I am so happy to be going through Unit 3 of AP Bio, and we're going to specifically be looking at topics 1 through 4. Okay, so this is about enzymes. First, we're going to look at metabolism. So metabolism is all the chemical reactions in an organism. And metabolic pathways, this is a really important concept about the series of chemicals reactions that either build up complex molecules or break them down. So with enzymes, you have a substrate. That substrate binds to the active site of the enzyme. There are intermediate stages where the enzyme is helping that substrate reach its transition state, and then there's a product. So there are two types of metabolic pathways: catabolic pathways and anabolic pathways. So catabolic pathways are pathways that release energy. So it breaks down complex molecules into simpler compounds. So if we think lots of complex bonds and everything in a compound, catabolic pathways break those down into smaller little molecules, and that releases energy because there's energy stored in that complex molecule. Think ATP. So it has adenosine triphosphate, and it breaks off one of those phosphate groups, and that releases energy that the cell can use. And it's now a simpler molecule. It's now phosphate and ADP. Anabolic pathways are the opposite of that. So if we now stop thinking about that and we start with this, we have lots of little molecules, and then we put them back together. So there's one, we bond it with this one, and then another one. This putting together into a now complex molecule requires that energy. So that is an anabolic pathway. We used energy to put that together. Think ATP synthesis. Think about the ATP synthase. That has to have a concentration gradient and H plus being pumped favorably in order to attach that phosphate back on. That's anabolic. Okay, so here's what I just said. So for catabolism, we have a complex molecule that's being broken down and releasing energy. And then the products of catabolism are used in anabolism to make more complex ones. Okay, let's keep moving. Energy. So energy is just a definition. It's the ability to do work. Organisms need to survive and function, and a loss of energy flow results in death. Okay, so there are multiple types of energy. There's kinetic energy, so that's running. There's thermal energy, so associated with the movement of atoms and molecules. So we know that lots of energy is released in non-usable energy is heat energy. So that's thermal energy. Potential energy is stored energy. So a ball at the top of a hill has potential energy. Once the ball's in motion down the hill, that's kinetic energy. And once the ball's at the bottom of the hill, it has less potential energy than it does here. Chemical energy, potential energy available for release in a chemical reaction. Think proton gradient. That has chemical potential energy because it has energy stored in one side, think of a membrane, and then it has the potential to be pumped favorably, say with an ATP synthase, right? Okay, so the laws of thermodynamics. These are really important for energy, for understanding the energy flow. So the study of energy transformations is thermodynamics, and it applies to the universe as a whole. So these are really comprehensive laws. So the first law is energy cannot be created or destroyed, but it can be transferred and transformed. So think of the way energy flows. So it comes from the sun, right? Autotrophs, like little flowers, they transform that energy through photosynthesis, through the light photons exciting the electrons, going through the photosystems, going through the Calvin cycle, making G3P. Do you see how that energy is going through a cycle? It's not being created. It's just being transformed from its original form into something, say, that the plant can use. And then once it's broken down through cellular respiration, it's used in cellular functions. and then it's eaten by a heterotroph. I'm going to attempt to draw something that would eat a flower. Okay. Maybe that's a anteater or something. I don't know. But the heterotrophs then eat the autotrophs, which did not create the energy. It transformed it from the photons. So see how that energy is moving up the levels and then back down, back down into the earth. See, all the energy comes from the original place. It's not being created by any of these. It's not being created by any of these autotrophs or heterotrophs, and it's not being destroyed either. It's just being transformed. Though lots of it is lost by heat, it's not being destroyed. So the second law is about entropy. Energy transformation increases the entropy and disorder of the universe. During energy transfers or transformations, some energy is unusable and lost as heat. So, see, the energy is not being destroyed, but it's being released through heat, which we're not using as energy. It's just being released as heat. So, cells create organized structures by taking in less organized starting materials. So how does this not violate the second law of thermodynamics, which says that energy transformations increase the disorder, but yet we are taking in little, small, less complex molecules and building it up into more complex molecules. So we're taking these and making them something like this. How is that not violating that it should be making it more disordered? However, after the squirrel ate the nut, catabolic reactions turn the complex molecules into simpler ones. So we're breaking them back down into simpler molecules again. So also we can think, look how complex this is. How is it? It looks like this is simpler and this is much, much more complex. When an entropy says that the molecule, as the energy transformation increases, that the disorder should go up and the complexity should go down. However, as the entropy of the organism decreases, so the entropy of the organism decreases, but the entropy of the universe increases. So through these even though these are complex, the entropy of the universe is still increasing. Okay, so now let's talk about free energy. So since the laws of thermodynamics apply to the universe as a whole, scientists use a concept called free energy to determine the likelihood of reactions. So that's what we want to look at, the likelihood of the reactions happening. So another key word here is energetically favorable. So when we're looking at reactions, we can tell if that reaction is going to happen spontaneously, and we'll touch on what spontaneous is in a second. Let's look at this formula real quick. So this is called Gibbs free energy. This is the total change in entropy, which we know from here, energy transformations should increase the entropy of the universe. Okay? And then we've got temperature in Kelvin and change in entropy here. Change in total energy here and entropy there. Sorry, my bad. Okay, so the free energy change in the reactions determine whether or not it occurs spontaneously. So spontaneity means that it will happen with no outside input of energy. So that means that if we just sat and looked at this reaction for 1 million years, it would happen. But that means that it will happen, but maybe it will take those million years. That's why enzymes are important and make sure that they do happen. So based on free energy changes, we can classify reactions as exergonic or endergonic. Exergonic means it releases energy. Endergonic means it takes in energy. So if you think, which one would be spontaneous? Something that releases energy or something that takes in energy? Okay, so if we think no outside input of energy, we would have to say exergonic, right? Okay, so free energy. Exergonic reactions are reactions that release energy. So cellular respiration, think. We have glucose, right? the first step of cellular respiration, we go ahead and break that into 2 pyruvate molecules, and that releases ATP, right? So it releases energy, and it breaks down a more complex molecule into 2 less complex molecules. That is exergonic and catabolic. So those go hand in hand, and the reaction is spontaneous. So let's look at the energy of the reactant. So glucose, look how complex it is. And it goes up to that transition state, and it falls down, and the product level is lower than the reactant level. That means that the reactants released energy because they had it stored in them. See how it's higher? That it had stored energy in there that released to get to the product level. So if we looked at endergonic reactions absorb energy. So photosynthesis, it takes in photons. So we'll draw this little leaf here. It takes in photons, which just excites electrons, and it ends up, after the Calvin cycle, making a molecule of G3P. So do you think in exciting electrons and transferring it to NADPH, that is less complex than G3P? Because I think it is. So the reactants have a much lower energy level than the products. So that's why it takes energy to do these reactions. It takes energy to store energy in the product. See, we have less stored in the reactants, so we have to take energy. We have to take energy and put it in the reactants to get the products having a higher energy level. See, it absorbs energy. Okay. So, remember how we said that if the energy flow stopped, it would result in death? Let's look at this now. Living cells have a constant flow of materials in and out of the membrane. They are not in an equilibrium. They need that constant energy flow and work to stay alive. So, cells perform 3 kinds of work: mechanical movement, transport, so think active and passive transport across the membrane, and chemical synthesis of molecules. So all molecules must be in motion. Some molecules have more energy than others. Think ATP, right? That has more than ADP. Okay? The molecule with lots of energy is kind of popping around. So think, if you ever took chemistry, you know that with heat, it puts energy into the system and makes the molecules move all around and hit each other more. So think of that as energy went into that and made the molecules speed up and kind of move around more. They have to be able to build up some materials more than others in order to do work. Okay, ATP. Okay, here we go. Adenosine triphosphate is the primary molecule that organisms use as source of energy to perform work. So active transport requires ATP to provide that energy for, say, something to be pumped across its proton gradient. So the structure is a 5-carbon ribose sugar and 3 phosphate groups. 1, 2, 3. This terminal phosphate group is the most important one probably in the body, right? Because this is where we get all our energy, in this little bond right there. ATP couples exergonic, so release of energy reactions, to endergonic reactions in order to do cellular work. So exergonic process, so when an exergonic one releases energy, this is energy being released. The endergonic process, which requires energy, takes this energy and is able to do its work, if that makes sense. So organisms obtain energy by breaking the bond. So if we go back up here, when this bond is broken, it releases energy. And that's because there's energy stored in that bond. It's this happens through a hydrolysis reaction. And hydrolysis reactions mean that water is added to break up the bond between the terminal phosphate and the other 2 phosphates. So the release of energy during hydrolysis from ATP comes from the chemical change. So the reactant of ATP has a higher energy level storage. It changes state to ADP whenever the terminal phosphate is broken off. And see how this difference in the level of ATP and ADP is? There's a difference in the level, which means that this is a release of energy when it changed from this to this. So in that breaking of the bond, the compound, the molecule, is changed from a higher energy to a lower energy, which is where this change is the energy released. Okay, so phosphorylation is when we take a phosphate and we put that on something else. An example of this happening is in the Calvin cycle, when we use ATP to store energy in that G3P molecule. We break off a phosphate group from the ATP, and that energy is released, and we phosphorylate the carbon molecules. Okay, so here is the hydrolysis of ATP. We break off that inorganic phosphate, and energy is released because of the state of this having more chemical energy stored in it than the new molecule made. Comes from the lowering of free energy, not from it just being bonded, not from phosphate just being bonded. It's from the lowering of free energy, where I drew here. See how this has more free energy stored? Now there is more free energy available to the cell to use. Okay, regeneration of ATP. So ATP is, so you know how it broke apart with the hydrolysis reaction? Then energy, it is broken apart to do work, and then ADP and a phosphate group can be recombined to make ATP again. So an example of this would be in the ATP synthase. We often see the proton gradient of H plus being used to phosphorylate ADP, so adding a phosphate group to it and making it ATP. So the rate of metabolic reactions. The laws of thermodynamics tell us if a reaction is spontaneous, but it does not describe the rate of a reaction. Okay, so we can find out if a reaction is spontaneous, but we don't know if it's fast or slow. Some spontaneous reactions happen so slowly, like how I said about 1 million years. We'll watch it and it'll happen, but it'll take 1 million years. So it would be impossible for our cells to take that full million years to do their work. So hydrolysis of sucrose and glucose, for example, would take nearly 1,000 years to happen by themselves. So how does our body use reactions that would take 1,000 years? Well, the answer is enzymes. Enzymes are macromolecules that speed up and catalyze reactions by lowering activation energy. So this is a spontaneous reaction, right? It has to go all the way up here to something called a transition state. Okay? And then enzymes, what they do is lower that amount of energy required to get to the transition state so that they can happen faster. They're not consumed by the reaction, so they can do the same reaction over and over again, and they're a type of protein. So if we remember some things about proteins, they have a special structure. They have 4 levels of protein structure. We have primary, secondary, tertiary, and quaternary. And if you bond to a protein, you can disrupt the hydrogen bonds or the disulfide bridges in the protein structure, and that leads to a change in shape. And if you have a change in shape, you have a change in function. So when something bonds to an enzyme, that can change its shape and change its function. Enzymes named ended ACE. So the enzyme that acts on a reaction called a substrate, that's a vocab word. Substrate, that's just what's going to go and bond to an enzyme. And the active site is where that substrate goes to bond. Okay? Enzyme function. So active site are open, the substrates go in. The substrates are held in an active site by weak interactions, and when they're all together inside the substrate, inside the enzyme with the substrates, this is called the enzyme substrate complex. That's another vocab word that would be good to know. Then, since the reactants are held together by the enzyme, they're able to reach their transition state easier, and they're converted into products, and then they're released. So induced fit, this makes sure that the substrate fits inside the enzyme well. So sometimes the enzyme changes its shape a little bit. See how it changed its shape a little bit here from here? To make sure that it's a perfect fit for the substrate and that the transition state can be reached. Enzyme catabolism. So remember what we said about catabolism. That is breaking down complex molecules into less complex molecules with the release of energy. So this breaks down complex molecules into less complex molecules. And anabolism, this builds complex molecules. So the opposite of catabolism, okay? Effects of enzymes. So enzymes are proteins, which means their 3D shape can be affected by different factors. Again, proteins can be denatured by temperature, pH, and chemicals. Change in shape means a change in function. So if the pH is super low, if an enzyme that does not belong in the stomach, so the stomach has a low pH. If an enzyme that does not belong there goes into the stomach, its 3D shape can be affected, and then there's a change in shape. So say it looks like this. It no longer can perform its function because it is denatured, and there's a change in shape, so there's a change in function. So there are optimal conditions. So temperature and pH must be optimal, and the rate of enzyme activity increases with temperature up to a certain point. So remember how I was saying about the molecules, when heat is put into the reaction, they start moving around faster and hitting each other and colliding because they're heated up, and the energy causes them to move faster and faster and collide, and that would make them react. So it's the same thing with enzymes. So when heat is added to the system, the energy is added to the substrate, which makes them combine more with the active site of enzymes that are ready to react with them. So, but after a point, the enzyme will denature because heat will break the hydrogen bonds in the secondary structure. Enzyme functions best at a specific pH. So we said pH can break the hydrogen bonds and change the function. So this is just a graph about temperatures. Okay, so cofactors. This is another vocab word that would be important to know. Cofactors are non-protein molecules that assist with enzyme function. So we can think magnesium, right? Magnesium, they can help with metals, magnesium, potassium can assist with enzyme function. So coenzymes, so organic cofactors can be vitamins, like vitamin B. So those can just help with enzyme function. Okay, so inhibitors. These reduce the activity of a specific enzyme. So why would we want to reduce the effectiveness of an enzyme? So think our body doesn't want to do more work than it has to, right? It doesn't want to waste energy, even though enzymes don't require energy, but it just doesn't want a surplus of a certain product when it doesn't need it. So say this enzyme could go all day. It could go for as long as you wanted it to, but the body doesn't want to have to manage an excess supply of this. So inhibitors would prevent the enzyme from making this certain substrate for a time to make sure that it's only doing its process when needed to. So permanent inhibitors, like toxins and poisons, can make the enzyme stop functioning forever. But reversible enzymes, that's what our body can regulate when it needs the enzyme to function. So this is just weak interactions. So competitive inhibitors, so these will reduce enzyme activity by blocking substrates directly from binding to the active site. And how would they do this? By binding to the active site themselves. So these can be reversed with increased substrate concentrations, and I'll show you about that. But basically, think we have an enzyme here that can bind perfectly we have a substrate that can bind perfectly to that substrate. Okay, substrate right to the enzyme. However, this competitive inhibitor looks just like it. So it binds to the enzyme active state, and that means that this goldfish-looking substrate can't bind there anymore, just because it's literally in its place. So this can be reversed if there's a lot of these goldfish-looking substrates, and that can mean that when the enzyme goes to bond with a substrate, it will more likely pick the goldfish one will be more likely to fill that spot rather than a competitive inhibitor. So just think, when there's more of the substrate than the competitive inhibitor, it'll be more likely that that substrate will bind with the enzyme and that the inhibition will be reversed. How about non-competitive inhibitors? So these bind to an area other than the active site, so we're always going to call that anything that's not the active site, we're going to call the allosteric site. So think, I've been saying, proteins, when something bonds to them, it changes their shape because of that protein structure, because of that hydrogen bonds and the tertiary structure. So we're going to think that is going to change the shape and change the function. So when something bonds to an enzyme, even if it's not the active site, that is going to change the shape and the function of the enzyme, so it can no longer bond to its specific substrate that it's supposed to. Its active site changed shapes. Do you see that? Because it is a protein. It is a protein, and it bonded to something else, which changed its shape, which changed its original function. So this is called non-competitive inhibitors, because this one was competitive because it bonded right to where the substrate wanted to go, the active site. This bonds to an allosteric site, which just changes the active site for the substrate. So a cell must be able to regulate its metabolic pathways, and this is how it uses its non-permanent inhibition. So control when and where active enzymes are active, and can switch genes that code for enzymes on and off. Allosteric enzymes have two binding sites, one active site and one allosteric site. So an allosteric site is any place that's not the active site. When something bonds to the allosteric site, it changes the shape of the active site. It could close or it could open it. So allosteric regulation. Molecules bind to an allosteric site, which changes the shape and function of the active site, which we've been talking about. It may result in inhibition or stimulation. So this means that the enzyme could be more open. It could mean that the enzyme will now accept the substrate. And now the active site, so say if the active site was like this before, and then an allosteric regulation binded to, then the body started, then there was something binded to the allosteric site. It now changed the shape to be this, something funky. So now that it's bonded to that, it changed the shape, and that's exactly the shape that the substrate would be. See, before this wouldn't fit, and now it does. So now the enzyme is open. So that means allosteric regulation isn't always for inhibition. It can also be for stimulation. So now it's able to accept that enzyme. The enzyme is able to accept the substrate when before it wasn't. Okay? So an activator is just what we were talking about. It allows the active site to remain open, but the inhibitor closes it. So do you see the difference? This is closed, then something binds to it, and now it's open to that substrate. Now it's open and something binds to it and it is closed. So this is just different ways that the body can regulate how enzymes function. So cooperativity, substrate binds to one active site on an enzyme which stabilizes the active form. Considered allosteric regulation, since it is binding to one site, changes the shape of the other sites. So binding to the active site, and then that allows the other active sites to remain open, whereas before they were closed. Feedback inhibition. So sometimes the end product of a metabolic pathway can act as an inhibitor to an early enzyme in the same pathway. So our body has really, really complex paths and metabolic pathways that take so many enzymes. Think glycolysis. There's so many enzymes that catalyze each reaction to take place. This means that, so at the very beginning of the pathway, we're going to call these all enzymes, and they're spitting out products that go to the next one, like that, and then this one spits out a triangle. Okay, so the body is saying, okay, man, I got a lot of triangles. This pathway has been working overtime. It is going to send one of these triangles back to the very first enzyme, and it's going to bond to it and inhibit the beginning of the pathway from starting. So now this enzyme is no longer going to make the stars. This enzyme does not have any stars to make the squares. This one doesn't have the squares to make the circles. So it's going to stop this whole pathway from functioning until these triangles have been used up by the cell, and then this triangle is going to be removed and it can start making stars again. So that is how the body can use the surplus that it gets from these pathways. It can use that to inhibit the very beginning and prevent this whole thing from functioning and regulate how much product it gets. So that's just really cool way that the body regulates itself. Okay, and this is a practice FRQ, and my answer is corrected if you want to go ahead and try that. And then here's another practice FRQ. Okay, so thank you so much for learning with me about AP Bio, Unit 3, Topics 1 through 4, all about enzymes.